So I'm Dr. Kashan Kasit again with a new video of the topic which is very important to the related to the cell, that is apoptosis. Normally our cell is performing different activities, metabolic processes in our body, but whenever there will be the external stimulus or injury to the cell death will occur by two processes. Number one is the apoptosis, the second one is necrosis. So here we are with the apoptosis. Apoptosis is a natural or programmed cell death which is under the control of the gene. So it is a normal phenomena. It is the normal phenomena of the body to engulf or digest the cells which are not wanted by our body. So it has its significant role in the removal of the unwanted cells. There are our 10 million cells, which are approximately 10 million cells which are forming inside our body every day, but these are dying again. So this cellular homeostasis is maintained by the process of apoptosis. The another importance is that whenever there will be the stimulus from the outside, which is either a toxin or a cell is under the attack of the virus or a foreign particle, the removal of the cell which is damaged and beyond repair. Actually every cell is damaged by the external stimulus, whether it is a bacteria or other particle like virus. So the cells which are not under the repair are removed by this process of the apoptosis. So how this process occurs, how the apoptosis is activated. I will give you the normal physiological examples of the apoptosis. In the development of the fetus, this is our hand and this is like separated, every finger is separated, but in the fetus it is wrapped like this. So this process of apoptosis is actually removing the tissues present in between the two fingers. So apoptosis is the process which is separating our fingers. The another example we will take here is the sex differentiation of the fetus, which is also degradation and regression of the duct system of the fetus, which is done through the process of apoptosis. So there are many other examples of the apoptosis. These are normal examples of the apoptosis. So activation of apoptosis, we were discussing how the apoptosis process is activated. Withdrawal of the positive signals and the arrival of the negative signals. What are the positive signals? Which are prolonging the life of the cell. The cell is prolonged and protected by these positive signals. Like positive signals are produced by the chemical stimulants like nerve growth factors, which is present in the neurons. Interleukin 2 is this one, interleukin 2, which is present in the lymphocytes. Whenever these positive signals are withdrawn, the process of apoptosis occurs. And the second one is the removal or arrival of the negative signals. The negative signals are either from the external or either from the death-gated ligand receptors. Death ligand receptors, I will discuss here the death ligand receptors. This is the cell membrane, normal cell membrane of the cell. It has some specific receptors for the binding of the death ligand. These death ligands are combining to the receptors DR5 and DR4, and the process of apoptosis begins. There are other negative signals which are external injury or the exposure to the radiations, ultraviolet rays. All these are the negative signals which are activating the process of apoptosis. Another example I will give here, the organelle, which is performing the process of apoptosis is mitochondria. Mitochondria is performing very vital role in the apoptosis because it is releasing the two proteins which are cytochrome C and the SMAC. SMAC is second mitochondrial derived activator of the caspases. What are caspases? Actually, the external or internal stimulus is whether external or which is either from outside or which is present inside. Whenever the stimulus reaches the mitochondria, activates the proteins called the caspases. Normally these caspases proteins are suppressed by the factor which is AIF, apoptosis inhibitory factor. So the cytochrome C is activating the caspases, and the SMAC when come in contact with AIF, these two then aggregate, and these then what? These also activate caspases. So these caspases are activated and the process of the apoptosis occurs. So next, what are the different morphological changes which occur in the cell during or after the process of apoptosis? The number one is the shrinkage of the cell. Actually, the cytoskeleton, which are the organelles present inside the cell, these are degraded by these caspases and the shrinkage of the cell occurs. The next one is the chromatin degradation. Chromatin is degraded and fragmented into the smaller particles. The next one is DNA is broken down. The cell membrane shows bubbled appearance. This cell membrane is also broken down. All the organelles present inside the cell are broken down and intracellular fragments are formed. These fragments are called apoptotic bodies. So this is how the process of apoptosis occurs in our body.